Bonsoir. Je suis enchantée de vous accueillir ce soir à l'Université de Genève pour notre événement intitulé « Le messager, une histoire de l'ARN messager ». Pour commencer, permettez-moi de me présenter. Mon nom est Martine Collard et je suis vice-rectrice de l'Université de Genève en charge de l'enseignement.
Good evening.
Je suis également, et depuis beaucoup plus longtemps d'ailleurs, biologiste et professeure à la faculté de médecine. Le laboratoire de recherche
I'm delighted to welcome you this evening to the University of Geneva for our event entitled The Messenger, A History of mRNA.
Un mécanisme essentiel au développement
A mechanism which is essential for development and the health of all living organisms, and which is not unrelated to the molecule we're going to be talking about at length this evening, messenger RNA. What is gene expression? Gene expression is the process by which, from the same DNA present in the nucleus of all the cells of an organism, to give rise to a multitude of different cells, blood, skin, lung, kidney, etc., are created, and then to ensure that they function according to the body's needs in the right place and at the right time. This mechanism, which is at the very heart of life, must be finely regulated, and that is precisely the role of messenger RNA. At the interface between the genetic code, which is housed in the cell nucleus, and the proteins which are responsible for executing this program, this molecule is responsible for giving the signal for protein synthesis and then rapidly degrading when its action is no longer required. In this respect, messenger RNA is absolutely essential to life and has been the focus of my research for over 30 years. During the COVID-19 pandemic, I was particularly moved when the messenger RNA-based vaccines were rolled out by the extent to which this molecule aroused fears and anxieties, and by how little was known about it. I was often asked by friends and by friends of friends just to explain what messenger RNA is and to point out that it's not a newly discovered molecule about which we still know very little. In fact, it had been studied for decades in contexts as diverse as oncology and muscular disorders. It's true that vaccines against COVID-19 have placed it in the spotlight, but its history, from the first studies to the Nobel Prize, is much more complex. That's why, together with Yelena Gantsov, the co-director of the film you're about to see this evening, we decided to organize a public screening to explain what messenger RNA is and to take another look at the extraordinary therapeutic hope that it inspires. After the screening of the film, experts will share with you the progress made in research and will answer your questions. This debate will be moderated by Isabelle Moncada, a journalist and producer with Radio Television Suisse's scientific and medical news magazine 36.9, which you all know, I'm sure. She has kindly agreed to moderate this discussion on a voluntary basis, and I would like to extend my warmest thanks to her. I'd also like to thank our experts, Professor Adrian Krainer, who has travelled from New York, especially for this evening, Professor Steve Pascolo from the University of Zurich, Professor Alexandra Calmy, who will be with us on Zoom, and Professors Mikaël Pittet and Arnaud Didierlaurent from the University of Geneva. I wish you all an excellent evening, and I will now give the floor to Yelena Gantsov, producer and co-director of the documentary The Messenger, A History of mRNA.
As we stand here together at the summit of 2024, it's striking how things seem much clearer than back in March 2020 when the world was a blur of uncertainty. And soon the conversation turned in the vaccination and the vaccine based on messenger RNA technology. And I must admit, like many of you, I had questions. I wasn't sure at all I wanted to get vaccinated. Although growing up in a medical family and once even considering going to a medical school, I decided to dive into research to better understand what stands behind this mRNA and who stands behind this innovation. Well, our journey of production, this film, was paved with challenges. And I think that my curiosity set the stage for what would become this film, The Messenger, a story of mRNA. The biggest challenge in the film was actually bridging the gap of the complexity of science and normal human beings. I will never forget driving in the car with my husband and trying to explain to him about the ribosome producing proteins and then DNA and RNA, and he said, wait a second. What is RNA in simple words? And I said, right. How can we explain this messenger? What is this messenger? Who is this guy? And we asked all the scientists in the interviews to explain how messenger RNA works. And the best explanation was actually given by Professor Steve Pascolo, who is with us tonight, who compared DNA to a saint book of life, like Santa. And I asked him, so, okay, if this is Santa, who are all the other personages around this DNA? Who are those little elves? And in a few days later, he sends me a drawing with a little kitchen and a little chef. And I look at this and I said, this is good. This is really good. But there was a little problem because there was a watermark across the whole page saying, not to share. And it's exactly what I wanted to do. I wanted to grab this drawing and share it with our animation team at Le Truc studio in Geneva. So I had to get the permission from Steve. And I must say that I think that this film is not just a science film. It's a human story of people who once decided to fight for what they believed in and to fight for their innovations. And I would like to thank from the bottom of my heart, to thank all the scientists who contributed to the film, to thank our co-producer, the RTS channel, my co-directors, Tarek Daoud and F. Duran, our composer, Jérôme Bauer, who is supposed to come as well here tonight, one of our editors, Kostas Makrinos, who is here, and all our technical team who helped us along this journey. So now let's buckle up and watch the film.
There is this concept that science is a lonely quest by a solitary genius, isolated from the world, frantically scribbling equations on a scrap of paper, and then shouting Eureka and running out onto the street to tell the world about the discovery. There's no question that phenomena like that happened in the history of science. But in truth, things that make a difference in the real world have features which are kind of hard to make a good movie out of.
The new strain of coronavirus was first detected in Wuhan City, China, over a month ago. There are now more than 220 confirmed cases.
We don't quite know how serious the disease is, how it's going to spread.
Now there are reports of human-to-human transmission.
It is 10 times more lethal than the seasonal flu.
The World Health Organization hit the alarm button, declaring coronavirus a pandemic.
We have rung the alarm bell loud and clear.
Le vaccin, c'est pour quand? C'est pour qui? Qu'est-ce que ça concerne exactement? Qu'est-ce qu'on sait précisément de ce que nous préparent les Moderna, les Pfizer?
We instantaneously understood that mRNA had really high potential.
On a été les premiers à vraiment croire en cette technologie.
I saw that mRNA is a technology which could be adaptable to our vision to develop vaccines.
Back then they were playing with all sorts of different technologies.
As soon as I got that vial, bang, everything worked.
So then we thought, well, this is interesting. Perhaps we can find ways of delivering the drugs.
And of course, immediately I offered him that I will make that RNA for you, and that's how we started.
A lot of people were very skeptical about that. You know, they didn't think it would work.
They take that technology, tweak it a little bit to make it work in the COVID vaccines.
We made major discoveries and we initiated a clinical trial.
The aim was very clear, to develop a vaccine as fast as possible.
The genetic COVID vaccines, they're not working.
Je crois qu'on peut applaudir. Hein.
I think we can applaud.
Félicitations à Jelena Ganshof et à ses collègues et à toute l'équipe qui a réalisé ce film. Je crois que je suis assez bien placée pour savoir quelle est la somme de travail.
Congratulations to Jelena and to those who made this movie. I think that it is a huge success.
On va laisser les
all the work that has to go into making such an interesting documentary.
Totalement mérité. Applaudissements totalement mérités. On va laisser les quelques personnes qui ne peuvent pas rester
now,
pour
some people have to go, so we will let them leave quietly, and I'm going to invite the guests to the round table come to the stage. So we have a very interesting perspective from different experts who are going to join us this evening. So first of all, we have Professor Adrian Krainer, who's come from afar, from the Cold Spring Harbor Laboratory. Please take a seat. So he's a biochemist and he lives in the United States. He is very renowned worldwide and he studies RNA gene splicing in genetic diseases and cancer. He's been doing that for years and he's described some of the medicine that is used for infantile myopathy that is used for preventing the death of children who die before the age of 2. It's called Spinraza, and we will talk about it in a second. And we will also talk about cancer and other diseases. Steve Pascolo, whom was in the movie, please come to the stage. So he did not come here on his bike, but by train from Zurich. Thank you very much. Trained in immunology at the Institut Pasteur. He worked at Tübingen in Germany and before coming to the University Hospital Zurich. Thank you to Switzerland. Thank you for Switzerland. And he also is a pioneer in mRNA and cancer, and he has worked on immunostimulation in cancer. We will come back to that. And Alexandra Calmy, professor, who is with us on Zoom. There you go. She is here, but remotely, because she's a doctor and she's responsible for HIV and AIDS here at the HUG local hospital. She works with Médecins Sans Frontières and she's interested in access to treatment and epidemics and immunity in particular. And she works on many clinical trials. Just like Steve Pascolo, she contributed to informing the population during the COVID crisis. So we saw her in the media, and she had to play the difficult role that we requested of journalists to intervene and talk to the population about what was going on. And here we have our next Mikaël Pittet, Professor Mikaël Pittet, who's from the University of Geneva, also an expert. Also renowned worldwide in immunology and cancer. He left to the United States for 15 years. He was at Harvard and then came back, luckily, and he wrote many articles and has made links between fundamental research and clinical research. And finally, we have his colleague, Arnaud Didierlaurent, who is also professor at the Pathology and Immunology Department unit. They actually work together. He is expert in biotechnology. He was trained in France, worked in the United States, in England, in research, and for GlaxoSmithKline for the development of vaccine adjuvants. He knows the processes very well, and he contributed to the commercialization of a vaccine. We will come back to it. So, Adrian Krainer, you come from afar. One question we have for you is, how do we go from the animal species to humans? And you've worked on deadly diseases, and has this helped you to work through clinical phases 1, 2, 3 in humans? Now, please take the mic. I understand French, but I don't speak it very well.
Passing from animal studies to studies in human, and so it's.
Vous l'entendez? Je ne sais pas s'il fonctionne. Alors, je pense qu'ils ne sont pas allumés. Merci. Heureusement qu'on a un ingénieur.
Maintenant.
Voilà, c'est bon. Je rigole. Je rigole.
Does this one work? No.
Voilà. Ils sont allumés? Parce que comme on est, comme l'intervention est filmée, il faut vraiment que tout le monde parle dans un micro, y compris vous quand on vous passera. D'accord, 30 secondes. Ah oui, moi j'en ai un qui fonctionne. Je pourrais vous le passer en fait. Oui, tout à l'heure, quand je vous passerai la parole, parce que l'idée c'est qu'à la fin de la table ronde, il y a un moment d'échange entre la salle et nous pour les questions. Je vous demanderai d'attendre d'avoir en fait le micro en main pour que, d'une part, Alexandra Calmy qui est à distance vous entende, et puis que d'autre part, sur la vidéo qui sera ensuite accessible au public sur le site, j'imagine, de l'université, qu'on entende en fait vos questions. En tout cas, merci d'être là nombreux ce soir pour nos intervenants et pour avoir vu cet excellent film. Ah oui, mais alors on pourrait commencer avec... Merci Martine Collard me fait signe, parce que Alexandra Calmy, en fait, on vous entend, vous n'avez pas besoin de micro.
Because you don't need a mic. So I'm going to ask you the questions I wanted to ask you earlier. If a new pandemic came along, would we be ready quicker than we were in 2020?
Thank you very much indeed for giving me the floor. We're lucky to have so many renowned experts on the panel. Will we be better prepared? Well, we can learn the lessons from the previous pandemic. During COVID, we were able to limit the number of victims thanks to the timely arrival of a vaccine, which was the subject of good quality clinical tests. Unfortunately, what we also observed was that we were not capable of actuating a solidarity mechanism in order to distribute this vaccine worldwide. There were various initiatives like the WHO COVAX initiative. They tried to collaborate worldwide in the field of vaccines with 20% of the receiving vaccines, but it was a failure for various reasons. There was a saturation of production capacity. This posed a problem. And there also was a degree of vaccination nationalism, which prevents redistribution of the doses, of course. Will we see that national vaccine nationalism in a future pandemic? Well, it's difficult to predict, but preparations are being made in the form of a treaty. Unfortunately, it was not signed last May at the WHO World Health Assembly, and it is running up against the barrier of the north-south polarization. I think we have to be extremely vigilant about solidarity and in the next pandemic, if there is one. We achieved a lot of things in COVID-19, but we did not make a good success of solidarity. How did you manage to do things in actual practice? Well, things were complicated as far as clinical tests are concerned. Now it's only we managed to do tests on 30, 40 or 50,000 patients in the clinical tests, which is an unprecedented scale and which gave rise to very fast results. I believe that was a success of clinical research, which subsequently was lacking, and for other reasons. What was difficult was coordination of the clinical trials in these consortiums. I think that in a future pandemic, I hope we will be able to improve coordination. Where do we stand? Question: where do we stand with regard to a vaccine against AIDS, against HIV deficiency? Answer: well, the mRNA vaccines offer several advantages, but we've been looking for vaccines against HIV for years and decades. We know the main reasons for this. It's the great genetic variability and the mutation of the HIV virus, which poses an obstacle to vaccines. Now, how could mRNA and that platform help us? Well, there are three firms which are sponsors of a currently ongoing clinical trial. We hope that will be successful. First of all, because we will probably succeed in preferentially stimulating cells which will put into production largely neutralizing antibodies, in other words, extremely robust antibodies, and that's what we need to protect people against HIV. This mRNA platform will be able to do that much better than what we had in the past. We've had many failures. The best vaccine that's ever been developed against HIV had a success rate of only 30%, so a lot of hopes have been pinned in this platform, mRNA platform. And even though the situation is currently promising, it's still a trial. So we don't know whether this platform will enable us to overcome the obstacles that are related to the virus itself. Please stay with us. Professor Calmy. Adrian, Professor Krainer, does your mic work now? Now, you have developed a therapy which protects motor neurons in babies. And does it enable you to, the body to produce what is lacking?
Ionis and Biogen is different. It doesn't use mRNA, but rather it targets mRNA to change it such that the cell can now produce a protein that's missing. And so for the work that we did, we use something called an antisense oligonucleotide, which is a short strand of RNA-like but chemically modified. But once it goes into the cells, it homes in on a particular messenger RNA that's made naturally, and it changes the way the cell handles it. And so mRNA can be a therapeutic in the case of a vaccine. It can also be a target for a therapeutic. And so there are mRNA, all genes make mRNA, and so there are different kinds. It's a very versatile type of molecule. And so the more we understand how it's made and how we can make it and how we can target it, the more potential there is for new kinds of medicines.
Est-ce que là encore, comme dans l'urgence de la pandémie, le fait que vous
just again, because you're working on a deadly disease, did that enable you to move from the animal to the human more quickly?
It wasn't so quick. I mean, it's relative. That takes years, so maybe 15 years from starting to work on the therapy until it could be given to a first patient in a clinical trial, and or including the clinical trials until approval. So it's a slow process, because when you develop a therapy, first you have to show that it has a high likelihood of working, but most importantly, do no harm. You have to make sure that it's safe. And so initially you do experiments in the lab to show that the mechanism is as you expect, or you can improve on it, you can optimize. And then there's a series of experiments that have to be done in animal models, typically in mice, to show that not only does it work in the test tube or in a cell culture, but it works in a living organism, and also that it's safe. So a lot of safety studies have to be done. And then in another species, again, before it goes into a patient already has a lot of problems. In the case of a lethal disease, you don't want to make it worse. And so after all of that, which takes a number of years, then you can start human studies very slowly, very carefully, if you have the luxury to do that. Small doses to ensure that it's safe in patients. And then when you have that confidence, you can increase the dose. So it's a process that normally takes a long time. Of course, vaccines is a bit different, but when there's a pandemic, there's an urgency, and so the timeline changes completely.
Alors, Steve Pascolo, merci. Steve Pascolo dans le cancer aussi.
I'll move on now to Steve Pascolo. In cancer as well, time is against the patient, and as we saw in the film, there is an enormous amount of expectation about medication, because current medications don't work, particularly for pancreatic cancer. Now, the question that's on everyone's lips is what is in the pipeline, as one says, and what is likely to be brought out onto the market soon. Answer: Moderna is in phase 3 for 3 types of cancer, for skin cancer and lung cancer. And one can hope that in 2 or 3 years' time, might be something. At the moment, the authorities ask us to prove that patients can survive longer. So you have to prove in a cohort that the survival rate of the patients exceeds the expected survival rate with customary medication. So we cannot proceed as we did with COVID-19, just to validate these therapies in the space of a few months. Moderna is now in phase 3. So you have to prove that the survival rate is longer for patients using mRNA than for patients without mRNA. Probably it would have to be at least 2 years, because the survival curve has to be sufficiently differentiated in order for us to be able to say statistically that it works. What we can say is that thanks to mRNA and its ease of production, what we have now are haute couture kind of customized vaccines made for each patient, and that enables us, well, with a photocopying machine, as it were, you can easily produce mRNA. So this enables you to produce tailored vaccines. In the past, we produced generic vaccines against cancer, but now, thanks to tailored, customized vaccines, we're in another different dimension, and we hope that in about 2 years' time, these vaccines will show their efficacy, especially for Moderna, against pancreatic cancer. So by about 2026-27, we should be able to come up with a new type of mRNA-based anti-cancer vaccine for breast cancer, pancreatic cancer. So we hope that each year we will have good news for patients. Mikaël Pittet. Now, this hyper-individualization is one of the objectives of immunotherapy. Now, as Steve Pascolo was saying, the fact that it's possible to quickly produce mRNAs, that enables you to thwart the resistances to cancer treatment. Yes, it's a great way of picking up on what Steve Pascolo was just saying. We are probably only 2 years away from being able to say what the impact of this type of treatment will be in the context of melanoma treatment. The acceleration comes from the fact that we have this possibility now of personalizing the treatment, and that is tomorrow's medicine. Thanks to the fact that we are understanding better and better the complexity of tumors and the ability that we have of somewhere being able to define the ID cards, as it were, of tumors or barcodes. One could use different ways of visualizing this. Now, when you think of the mRNA approach, in principle, we have the possibility of being able, relatively quickly, at least on paper, of doing this personalization. So this means that we will see a fairly incredible evolution in the coming years. And the tests that are being done in the field of melanomas should produce positive results, particularly as regards the signals that are coming out. One has to be a bit more cautious when it comes to pancreatic cancer, but in every case we know that we are all on a new treatment track, which will probably move forward much faster than in the past, and which will enable treatment to be personalized. And that's going to interest us a great deal and change lives for many patients. Question: you draw a distinction between cancer treatment and cancer vaccines. There may be two different approaches. Answer: well, the term vaccine, when used for cancer, is rather complicated because we talk about a therapeutic vaccine, which is maybe a contradiction in itself. There is a second mic, in fact. And we're talking about vaccines because basically in this context of cancer, we try to stimulate an immune response against or to the tumor. And that's what we try to do in the case of infectious diseases. We try to induce response to the pathogen. That's why we call it vaccine, but we're very close to a therapy. Question: but it's a mutation of the virus and of the cancer cells. You can come back to the patient with this. Yes, that's an advantage of this technology. We can quickly change the code and adapt depending on the evolution of the cancer or the pathogen. Question: so when cancer becomes resistant to chemotherapy, there is no longer any outcome. Does this mRNA technology mean that there will always be a possibility in future of not finding ourselves in a therapeutic cul-de-sac? Answer: today we are realizing that for a tumor to develop and become macroscopic and have a real negative impact on the patient, this means it has in some way escaped the immune system. Now, our immune system is really capable of eliminating cancerous cells in the course of our lives without us realizing. The mRNA vaccine or therapy has the possibility of generating very strong immune responses which are directed against the tumor. So here we can even say that depending on the mapping of the tumor, we can either target particular features of the tumor or other elements of the tumor which may be more generic and which we might find in several different patients in order to generate a strong response. And that's the ability of these mRNA-based approaches to stimulating the curiosity of therapists working in this field. I have a question to put to Adrian Krainer. Now, in cancers where you are carriers of a mutation, I'm thinking of breast cancer with BRCA1 and BRCA2 mutations. In a technology like the one used for myotrophy of spina, could that serve as a preventive vaccine?
In that case, maybe it's not a vaccine, but we could, and we try to do these things to introduce antisense oligonucleotides targeting the mutant mRNA that the cancer cell depends on. And so there are many experiments and even clinical trials based on that. The issue is that, as you mentioned before, the cancer cell learns to escape that. And also the issue is you may be able to kill a cancer cell that way, but if you don't kill 100% of the cancer cells, then the remaining ones will continue to grow. And so that's a problem of how to reach all the cancer cells. With the vaccine approach, if you use the immune system, and so you're not targeting the cancer cells directly, but you enable the immune system to target the cancer cells. I think in practice, cancer is very tough, and so in practice you may have to combine two or more therapeutic approaches to target the same cancer, and so you get a kind of a synergy. But I don't know, time will tell. Would you want that?
Arnaud Didierlaurent, ouais. Oui, je voulais juste peut-être ajouter une autre approche qui est également testée actuellement.
Arnaud Didierlaurent would like to refer to another approach. Thanks to technology, it's possible to bring in inflammatory molecules into the tumor. That's when you directly inject the RNA into the tumor, you generate a local inflammation, which gives rise to the response. Moderna has a program on this at the moment. It's also a way of solving this problem of having the right cells which go to the level of the tumor. This is also an interesting approach, quite apart from the need to stimulate the immune system. Steve Pascolo, you say that in the space of a weekend you can produce the mRNA, which is necessary to solve a problem. Does that mean that you have to relocate the manufacture of vaccines or therapies, and that we will need many more biologists at the local level? We will need more local knowledge when you get away from this industrial vision of having the same therapy for everyone, with which you irrigate the whole planet. Now, sometimes we have shortages of medicines. Answer: well, there is automation of robots, RNA printers maybe, could produce RNA locally. There are several industries in USA and Europe which are moving more into automatons, the size of a TV set, which can produce RNA on the spot, on site, on demand. And that will probably account for a large proportion of RNA production, and that will reduce costs and speed up availability of these vaccines. As regards innovation, there's also innovation on the current technologies which will be robotized, and new technologies which will be even faster and cheaper. I just wanted to add something about the various possibilities of RNA. Intratumoral injection. In medicine, RNA is a kind of Swiss Army knife. You can do several things: vaccines, therapies. You can also do anti-cancer cells. In other words, you modify the blood cells with RNA to produce tumoral killers. So there really are various aspects. You can inject into tumors. You can create vaccines. So with mRNA systems, we have a toolbox which enables us to do a lot of things, and which creates a lot of hope for patients, in particular cancer patients. In Yelena's film, a question, we saw the importance of these capsulated solutions. We saw that there are startups and also a lot of large big pharma companies working on this. Now, Professor Calmy was talking about the mention was made of the difficulty of transporting these unstable vaccines to hot parts of the world because they have to be refrigerated, and this restricts the possibility of transporting them. Answer from Steve Pascolo: that's no longer the case. Steve Pascolo. Well, because of the lipidic structure, initially they had to be vaccines had to be kept at -80. But now a Californian company has come up with a lyophilized vaccine. It's freeze-dried, so it's stored at ambient temperature. Moderna has a new lipid formulation which is stable in a liquid at ambient temperature. So some innovations have already been made and which overcome the cold storage problem. So they're stable at ambient temperature, so you can store them at ambient temperature, and this improves the prospects for distribution. Question: we saw in the film mention of the patent disputes. Of course, some people are in favor of patents, others are against them, and the possibility of having to have money to get access to these vaccines. And there are also fashion effects. Professor Didierlaurent, are the old technologies already obsolete, or is it worth investing energy and money in the old therapies. Professor Didierlaurent, the question is, will it replace all vaccines? I don't think so. I think that currently today's vaccines which are administered to children are very safe. At the end of the day, I think that these vaccines do the trick. They perform their role. But in some applications, well, in a pandemic, obviously you need a technology which has to go fast in order to produce vaccines, as Alexandra mentioned. And in some cases, we see that, if I take the example of flu, a lot of developments are ongoing for flu vaccines. The first RNA-based vaccines showed that they were more or less equivalent to traditional conventional vaccines, but now there are some data which show that they are superior to conventional vaccines. This shows that technology cannot necessarily replace existing methods for targets which traditionally were difficult to reach or to resolve. But will they replace conventional flu vaccines? Because the problem is that the viruses mutate so quickly that prevents effectiveness. Professor Didierlaurent, well, the seasonal influenza does not vary as much as that. Work is done each year to define the strain, but it could be a relevant technology for the future, but I think that the added value involves making combinations. Today, many companies are working on a vaccine which would combine a COVID vaccine and a flu vaccine, because you can combine the RNA. So, and there's also work being done on a virus which causes bronchiolitis in children and young people. So ideally there will be just one vaccine which will be injected at this time of year to protect us for the rest of the winter. So in that context, maybe the traditional conventional vaccines might be replaced by new ones. Adrian Krainer, one of the questions I have about patents is the cost of these treatments and therapies and access to them. And in rare diseases, the price is even more of an obstacle. How do you envisage the future in this field? We see that the healthcare systems, even of rich countries, have almost bumping up against the limits of what they can afford with an aging population and ever more expensive vaccines and cures.
Every country is a little bit different. I'm also not a big fan of patents, but the reality is that without patents, there is no investment in research and development, and so it's a mechanism that works. Nobody's happy about it, but the system produces efficacious, effective drugs and therapies, and there is a problem of access and equity, of course. But I don't know what the solution is, but patents, it costs a lot of money to file patents and maintain them. And so companies don't do patents in every country. They do them in the major markets where they can obtain a profit, and they use that profit for new research and development. So they will patent in the US or North America, Europe, Japan, those are major markets. They won't patent in other countries, so in principle, those countries or companies that go to those countries can manufacture the same drug without having to worry about patents. In practice, how much does that happen? I don't know. Then you have the issue is the patents have a limited life, right? Let's say 20 years, and some of that time the clock is running during the clinical trials and so on. And it has a perverse effect, because after the patent runs out, then anybody is free to make that drug, and maybe it can be cheaper and there's more access to it. Of course, during that time, there's a lot of markets where there is no access. That's very tragic. But at the same time, the companies know they can only make a profit during, 15 years. Therefore, they charge a lot more, because that's the window of time during which they can recover their investments. So it's very complex. I don't know if there's a solution, and is there a solution for the whole world, ideally there should be, but I don't know what it is.
Alexandra Calmy, vous vouliez intervenir?
Alexandra Calmy, would you like to come in here? Yes, I would. This is a very important point. We saw this very acutely during the VIH pandemic. And in Geneva, we saw a generic competition. One point is very important. With this personalized method, which we are going to see arriving on this mRNA platform, this Swiss Army knife, as it's been referred to, and rightly so. What bothers me is the concentration of the manufacturing capacities. In other words, are we going to be capable of decentralizing, or are we going to stay with very limited production capacities, which mean that even if it is a personalizable medicine, maybe we will only be able to personalize it in extremely expert centers. At the moment, there is a tech transfer center specifically for mRNA in South Africa. That's the kind of effort that rich countries like Switzerland, with all its capacity, which is not only academic but industrial, rich countries could participate maybe more visibly in this type of development, so that these platforms and this technology transfer can be decentralized more. I believe that's an area in which our country could have a substantial impact, and that's a point I would like to underscore today. We need to contribute to the ability to decentralize these mRNA messenger production platforms. Well, that's your field, is it not, Professor Didierlaurent? I would just like to add a couple of points to what Alexandra just said. You need expertise to make a vaccine. You need a certain amount of expertise. We have to develop this expertise in the countries you referred to. It's not just important to set up factories, but to bring in the expertise. That's important. One of the problems is that are the vaccines that we're proposing adapted to each country? For example, in the case of Africa, COVID as opposed to epidemics like malaria and tuberculosis, the question is which vaccine has priority. So an mRNA vaccine is being developed for malaria. So this has to be taken into account. Question: is the risk not? Isn't the risk that we will only see innovation in the areas which are profitable and startups take risks? You experienced this yourself, Steve Pascolo. How can one innovate without always being faced with this question of profitability afterwards down the line? Steve Pascolo. There are a lot of developments. There are European groups which are developing vaccines against malaria. The European Union and Switzerland are participating a lot. Moderators, so it's not just BioNTech and Moderna. No, there are many companies which are being funded to develop vaccines which will be used in lower income countries. Now, as regards automation and solutions of the future, one cannot envision the future with today's solutions. In future, we will have more automatons and robots which will make possible local production without that much expertise. Maybe an expert in Switzerland will be able to order a robot, an automated system which would be able to work in Nigeria. So AI and robotization will eventually allow global access to therapies. The poor countries will have to wait a bit. But thanks to automation, not only our countries but other countries will be able to have access to mRNA-based solutions. The question, it's also a matter of providing work to young biologists and similar people. Now, the pair of AI and mRNA, is this not a rocket? Answer, well, yes, one could imagine that it might be version 3, 4, or 5 of the rocket that we saw at the end of the film. all these considerations are exciting, but the terrible reality is that one man in every 2 and 1 woman in every 3 will suffer from cancer in the course of their lives, and the number of cancer patients will double in the next 20 years. It's a global problem, but at the same time it's also a local problem. So there is hope out there, thanks to these technologies, hope of being able to do much better than what we're doing today. But all these challenges which we are addressing this evening extend over and beyond the problem of understanding the utilization of the molecule. And all these questions are of capital importance, and I feel very badly placed because these questions are far removed from my field of expertise, although they are extremely exciting issues. Moderator, there's another question which arises when we're talking about this technology, which gives a lot of people hope, apart from treating people in pandemics and people suffering from cancer, that is, people suffering from brain diseases. Maybe Adrian Krainer will answer this question. What is the hope that's out there for those people? Is it important to clean up amyloid proteins at an early stage? Is it important to target the substances which destroy our brains?
I think a lot more basic studies have to be done to understand these diseases better and to better understand what to target. I mean, the tools exist, and mRNA may be one of them, and mRNA vaccines may be another, and targeting mRNAs in cells, that's being actively investigated for Huntington's, for ALS, Lou Gehrig's disease.
Et même pour le vieillissement en général, on utilise l'ARN pour essayer de rallonger les télomères de nos cellules pour contrer le vieillissement naturel.
And that's a double-edged sword, right? Because the telomeres, yes, it has to do with aging, but it also has to do with cancer. And so if you go too far one way, you generate one problem. So, you have to find the right path for that. So, of course, all of these technologies can contribute to these diseases. And we're also learning how to deliver these types of therapies to the brain. And so that's, there's a lot of progress there. There's a lot of room for improvement. So I would say, yeah, there's hope. But these have to be pursued both with the conventional approaches and with these newer approaches. And so I think there will be successes, not in the distant future, I hope.
Vouliez compléter?
Would you like to add something? Yes, maybe an example. When you are trying to treat a viral infection, when you're talking about multiple sclerosis, for example, when you create a vaccine which which protects us from multiple sclerosis, potentially you can reduce the number of multiple sclerosis. That's also the case for type 1 diabetes, by identifying the children who are likely to react to a virus, which targets the virus. Yes, especially children who are at risk of reinfection. We have proof of concept for infections, and there we can have an impact on long-term chronic diseases, particularly in elderly people. One can imagine that in future we will have a whole battery of vaccines or therapies which will enable us to improve the overall general health of elderly people and reduce the incidence of these chronic diseases thanks to our immune system, about which not so much was known until recently. Now, it's difficult to get through to the brain from the blood, but people receive oligonucleotides, which is directly injected through the Rachidian canal. It's not very pleasant, but there are lots of clinical studies on this. This can be an advantage because there you can inject relatively moderate doses, which will be in the cephalorachidine liquid, in the spinal fluid. If they're directly injected in the central nervous system, the spinal fluid, they can be effective. And this can be advantageous in that you have a volume which is smaller. So if you inject an RNA, which enables you to prevent development of the plates, sclerosis, you're in a more enclosed system, and there you carry out the injection only once a month, even though the actual administration is unpleasant. Moderator, and then there's a risk of inflammation. Yes, but people are normally on corticosteroids when they receive these injections. There's also an advantage with regard to approaches against brain diseases. The volume is more restricted, and that's why quite a lot of clinical tests are being conducted with oligonucleotides in order to combat degenerative brain diseases, Moderator. I now turn to the audience. Does anyone in the audience have any questions or comments? I know some students are in the audience. Adrian Krainer, you're in charge of the mic for the guest. I have difficulty in seeing you. Good evening, madam. Thank you very much indeed for participating in this debate. Thank you for contributing your scientific know-how. I have one or two questions about the side effects. In your opinion, are there side effects which are generated by this technology concerning mRNA, messenger RNA for COVID, even though the molecule is very well known? 30 years ago, studies were carried out on cancer in connection with this. We got to know this molecule thanks to COVID, and we know it will affect receptors in the myocard and cerebrally. What are the side effects, if there are any? Because they are not harmless. I believe there are many people who have strokes. Quite a lot of people have had strokes due to anti-COVID vaccinations, and there are also some scientific articles which have shown that cancer has been caused by these anti-COVID injections and a whole series of degenerative diseases which are accumulating. So, other verifiable second side effects? Answer from Steve. Pascolo, there are some known side effects, myocarditis in young men. Pharmacovigilance has never been as strong as against these vaccines, the COVID vaccines. No vaccine has ever been studied as much as the COVID vaccines to find myocarditis. It's really like looking for a needle in a haystack. But all the countries, Northern Europe, Central Europe, North America, all the agencies, all health agencies followed very carefully all the populations who were being vaccinated because they wanted to be absolutely sure about vaccination security. So there is what you mentioned, myocarditis, which affects young men, but so no vaccine has been scrutinized as much as the COVID-19 vaccines. But there was also a SARS-2 epidemic. Now, how can people dissociate between the effect of the vaccine and something else? Of course, people tend to say it was the fault of the vaccine, but it may have been the fault of an infection which they contracted. So I think in the whole history of medicine, no vaccine has been subject to as much pharmacovigilance as the COVID-19 vaccines did. The vaccines have been scrutinized over the last 3 years by different health agencies, and they function in different ways in France, Finland, US. Each one functions in a different way. Each agency has different methods and different approaches, and they all scrutinize the vaccines. If there really had been something other than myocarditis, we would have known it. Alexandra Calmy, Professor Calmy, would you like to answer? Professor Calmy, online. I share 100% the opinion of my colleague. There was a lot of pharmacovigilance at the national and international level. There are the side effects. There is the side effect which was mentioned. there are some side effects to the COVID vaccines, but you have to realize that just as for thrombosis, it's the thrombosis that gives rise to myocarditis. A French report found that the risk of having a myocarditis is 7 times higher when you are sick with COVID than when you just received the vaccine. So I think you also have to consider this in the context of the illness that we want to protect against. There are some side effects, but I believe they are being followed up on very transparently together with the whole community. And I think this kind of public debate is very important because it's important to show what we know already and what we are going to learn in addition in future. Is there another question elsewhere in the room? What I would like to know is the following. Despite everything that you've said, does the fact that we are being vaccinated not turn us into sheep? Do we know what the repercussions might be in 2 or 3 generations' time? We know that in the past, mutants, and it turns us into mutants. Well, no, these RNA vaccines don't turn us into mutants. When you're affected by SARS-CoV-2, you receive RNA in a capsule, and that's going to multiply in your whole body. When you're affected by SARS-CoV-2, you're going to have RNA also for the spike, but also many other RNAs for the other proteins of the virus, which will be in the whole of your body from tip to toe. So it will just be a locally administered dose. But in your life, you are constantly exposed to viral mRNAs in the form of colds, like the cold that I have at the moment, which you may have later. These you have the virus from this mRNA almost the whole winter, which develops in your lungs and skin. This does not mean that you will become transgenic. This RNA is absolutely everywhere, in everything that you are drinking and eating, and also in the viruses that affect you. There's an incredible quantity of RNA which is produced when you are suffering from SARS-CoV-2. Moderator, as regards the question of adjuvants, it's the question of adjuvants in these new vaccines. Could you say something about adjuvants? Steve Pascolo, traditionally adjuvants are aluminium. Now there are new saponin-based adjuvants, but that's why I don't really agree with Arnaud. I think that RNA systems will replace these adjuvants in future. They're also vegan. They're also vegan. And some vaccines have an impact on eggs and hens, but people who are allergic to chickens can take these new vaccines. You need 10,000 liters of culture to make a vaccine. So RNA ticks all the boxes, even though it's not a matter of priority, it's not urgent. But in the long term, RNA-based systems have advantages compared with conventional existing systems. But thank you for asking this question. It's important to know that RNA is everywhere. It's in this vaccine, but it's also in viruses. And that's why it needs to be followed. At the moment, you can say that RNA vaccines and viruses don't make people transgenic. You're defending hens. Is that it? Well, I just wanted to add, says Professor Didierlaurent, we have to understand how the viruses work in order to create vaccines. It's not just an RNA, a technology. So that's why we have to continue to do fundamental research, because vaccines don't just drop out of the sky. Moderator, but the fact that we have this promising technology, does this prevent us from going and looking elsewhere? Answer, no, on the contrary, I think this will speed up the development of vaccines. We can quickly get a proof of concept in humans. We've seen that they can be used safely. That's going to enable us to come up with proofs of concept and to test several approaches quickly. And maybe in a more distant future, this is going to be a technology which will become important. But currently used vaccines will not be replaced by RNA anytime soon. I agree with you on that. Question from the audience. At the molecular level, sorry, I'm rather stressed. At the molecular level, what does tailored treatment mean? Which RNA is injected, and how do you tell whether there is a modification, a mutation? How can you modify the treatment in order to overcome the mutation? Professor Pittet, I can't see you directly in the audience there, but to answer your question, one idea would be that if you are able to get a small piece of the tumor, that will enable you to get access to determinants which are specific to that tumor. So if you take patient A, if you take a little piece of patient A's tumor, you find things which are unique to that tumor, and thanks to that we will be able to develop a drug, a medicine which will be capable of recognizing those specific elements. And if you repeat the same exercise with a little piece of tumor from patient B, there you will have other different determinants which will enable you to produce messenger RNAs which will be different. So one way of doing personalization is, for example, to go and locate the patient's tumor, to analyze it in a test tube in vitro, to analyze the tumor. If I can just pick up on what was said earlier, it's true that these studies which are done in humans are extremely important. That's where we want to head, but it's still a matter of experimentation, and experimentation on animals is still important. For example, it's conceivable that in future, the next study on a type of study will prove that we have a good immune response, but we don't have good efficacy against the tumor. Why? Because we may well have lymphocytes which are produced, but they are not able to go into the tumor. That's one example among many others. We have to understand this, and we have to be able to control it. So it really is an ecosystem of clinical and experimental studies which have to go hand in hand. Moderator, yes, states have to find the juste milieu between investment in fundamental research and clinical research. And then there's the question of the damage which is caused by our environment. Steve Pascolo, these RNA vaccines also benefit from sequencing. There is one bit of the tumor which is sequenced. The genome is sequenced. It's the benchmark genome. And with the two references, that of the tumor and the blood, you can see the mutations which are specific to the tumor. And then you produce a synthetic DNA which will code for little peptides, where in the middle you have the mutation. And you create an mRNA from that, and that will code for little peptides with 21 residues in the middle of the mutation. The immune system will recognize them and then will recognize the tumor. So in terms of logistics, it's rather complicated because you have to send a bit of the tumor and some blood into this sequencing. It takes about 1 month to get the whole sequence, maybe a bit less than 1 month now. And then from that, among the dozens or hundreds of mutations, you select some of them, some of which are more relevant clinically. Then you do the DNA which codes for this peptide with these mutations, and then you make the RNA. It's rather complicated, but that's how we proceed for clinical studies. Last question. Thank you for your participation in this debate. You talked about haute couture for therapeutic vaccines. That makes us dream about the therapeutic potential, but at the same time, someone mentioned caution with development, the need not to move forward too quickly. My question is, how can one reconcile tailored treatments, which have never really been tried out, and with the need to move forward quickly? How can one reconcile these conflicting goals, personalization and coming up with proof? Well, bone marrow grafts are very individual treatments which have been done pretty quickly, which are permanently available. So one has to realize that this type of attitude for highly deleterious diseases, well, there are precedents for that. We are not working on unknown territory. Bone marrow grafts are individualized treatments which have been put in place without having the need for any large-scale studies. RNA technology today is, we can base ourselves on current knowledge thanks to large-scale deployment of COVID vaccines, particularly with regard to certain aspects of safety. Now, we don't want there to be too much inflammation in some cases. We can verify that, but in any case, we don't want to go too fast. It's important to demonstrate safety. We cannot ignore that. In some cases, of course, we would like to go faster, but at the same time, it's important that these RNA vaccines remain safe. And we have to do things the way they have been requested to be done by the regulatory authorities. And that's how, in the end, we will come up with something safe and worthwhile. Alexandra Calmy, do you wish to add something? Yes, this point is indeed important. What pandemics teach us is the absolute necessity to collaborate together, to work together. We saw this for HIV, we saw this for COVID, we've seen it also for mpox. It's absolutely necessary for the community to stay close to the health community, health professionals, and also politicians and regulators. So that will enable us to reconcile the need for safety and at the same time the need to move forward quickly. We will not solve these problems on our own in a corner. And by collaborating better together, we will be able to reconcile these conflicting requirements. Adrian Krainer, you have the last word. Alexandra Calmy talked about politics. What about the future of biomedical research in the USA? Are you going to ask for political asylum in Switzerland?
We're going to have to see what happens. I mean, science will survive, but I think that we'll have some hard times because, well, part of it depends on, the public has to believe in the integrity of science and drug development. We depend on the public, for support through, in the US, there's the National Institutes of Health. I think the next 4 years are going to be tough, but life will go on. We're just going to have to see. Yeah, so I'm worried. Yeah.
On imagine. Merci beaucoup. Merci à vous cinq d'avoir participé à cette table ronde.
Thank you, all five of you, for taking part in this round table, even though it's always frustrating because we would have liked to be able to say more. The purpose of Jelena Gantsov's film is that it should be seen by as many people as possible, so as to create a dialogue between the world of science and society. She came along with another colleague, and their aim is to foster a dialogue between students, the public at large, and researchers. And if it interests you, you can go on the website insidetomorrow.media or have a discussion with Yevgenia, who is here behind this camera at the end of this evening's event. Thank you very much indeed for your questions, and we wish you all the best for the future wherever you come from. Thank you also to the technicians and the interpreters who've enabled us to have this dialogue with Alexandra Calmy. See you soon. Thank you.